So I'll be talking about the molecular mechanisms of altered GERD channel signaling in GNB1 encephalopathy and specific GERD drugs as a potential therapy. So GNB1 encephalopathy is a rare neurological disorder that's recently discovered in 2016. And it is characterized by many symptoms like general developmental delay, epileptic EEG, or many different kinds of seizures, and many other symptoms. So all these effects takes place ever since the patient is as an embryo. So we could target other symptoms probably through gene therapy, but for seizures or epilepsy, we could target them using drugs, and we could alleviate the symptoms and increase the quality of life of the patients. So our objective is to alleviate the epileptic symptoms. So generally epilepsy is mostly because of dysfunction in ion channels or receptors. So if you look at the signaling cascade for G proteins, mainly the beta gamma. So beta gamma is an obligate dimer. Beta has 5 isoforms and gamma has 12 isoforms. And they directly interact and regulate many effectors like ion channels and enzymes. So what could go wrong if there is a mutation in G beta? So it could be either the interaction between the G proteins and the GPCRs could be disrupted, or the interaction between the gamma and the beta could be disrupted, or the protein itself could either underexpress or overexpress and leads to the loss of function or gain of function of the effectors. So if you look at the gene, there are so many exons, but these mutations mostly happening in the exon 6 and 7. This is the region which generally codes for the interface where it interacts with the either effectors or G alpha protein. Out of these mutations, we chose 3 mutations. This one was because during that time, the first mouse model with this mutation was being characterized. And the IITT is because the first patient that was diagnosed in Israel is with this mutation, and the ITN is just another variation of the same position. So in the structural studies, we saw the IITT comes in contact with the channel, but not the IIT position. When we look at the binding affinity, except for GERK1, in K78R mutation, everything else is normal. We also looked at the interaction between the G proteins and the GPCR, and also the dissociation and association of alpha and beta gamma, and the binding of beta to the effector. Here is the GRK, to see if every step of this cascade is okay, or if it's being affected by these mutations. And we see none of the mutations affect any of these steps. We also looked at the voltage-gated calcium channels. Beta gammas they inhibit the voltage-gated calcium channels, and none of these mutations had any effect on the inhibition on calcium channels. They are similar to wild type. Even their kinetics are similar to wild type. So next possible suspect is the GERG channel. This is a classical beta gamma effector. So GERG channels are activated by the beta gamma, and functional GERG channels are tetrameric ion channels. They generally mediate the inhibitory G protein mediated neurotransmission in the brain, and they are also implicated in many disorders, in addiction and many others. So as I said, a functional GERG channel is a tetramer. It could be either the heterotetramer with 2 different subunits, or homotetramer. And 4 G beta gamma complexes are needed for the channel to be activated. And if you look at the diversity and the expression, in brain specifically 1, 2, and 3 are expressed, and in heart it's 1 and 4, and in glands like adrenal gland, again 1 and 4. These patients show only neurological symptoms, so we focus only on neuronal GERK channels. So we start with the K78R mutation. So we did a dose response of beta gamma with this is with the GERK1-2 channel. It's a heterotetramer. So this is the dose response, and the black line is the wild type and the red is the mutant one. We could see there is a gain of function at the lower dose, but the loss of function at higher dose. These are the currents. And simultaneously, when we looked at the expression of the beta gamma in the membrane, so we saw consistent increase in the expression compared to the wild type at all the doses. When we look at the expression of the channel in the membrane at different doses of the beta gamma, we could see there is a decrease in the channel expression in the membrane. All these experiments are done in like xenopus oocytes. It's a heterologous expression system. With GERK2 homomer, we saw only gain of function effect. There's a higher expression at all doses, but we didn't see any loss of function effect. So K78R has a dual effect on GERK1-2 channel and just gain of function effect on GERK2 channel. We also performed single channel recordings. We used 2 different doses because if you see here, so it has a maximum activation at 0.5 and like loss of function at like 5. So we chose those 2 doses, and we saw there's a decrease in P open at both doses. This is a cartoon just trying to explain the dual effect of K78R. It's based on the graded contribution model done by Jakubowicz from our lab. These are like a number of beta gamma molecules, and this staircase, it shows the contribution of G beta gamma bound contribution of G beta gamma to the P open of the channel. So these are like 4 beta gamma binding sites in the channel. So if there is a 1 beta gamma bound to the channel, this is the P open. So assuming this is like 0.5 nanogram of K78R, if we look at the previous one. So here the expression is much better than the wild type. So at this case, so all this is the endogenous beta gamma. So if at this 0.5 nanograms, even one molecule of K78R goes and binds to the 4th positions, there is an increase in current, but not to the maximum extent if there was a wild type beta gamma. But at 5 nanograms, all the endogenous beta gamma, wild type beta gamma was replaced by the K78R. And when all 4 sites are bound by K78R, it's a loss of function. So at single channel level, K78R is a loss of function mutant. At whole cell level, it's a gain of function mutant because of its overexpression. Because it's a gain of, we also performed in the presence of M2R just to make sure that physiological conditions what's the effect. It's a gain of function effect in the presence of receptor also. We used ethosuximide, which was known as a T-type channel blocker, which also blocks GERK with much less doses. It has higher sensitivity. And we saw at the therapeutic dose, ethosuximide blocks the currents, the hyperactivity of K78R in GERK1-2 as well as GERK2 channels. IATN and IATT, so these are loss of function for GERK2 channels and GERK1-3 channels, but not for GERK1-2 channels. We also increased the expression from 5 to 10. We doubled the dose of the RNA and tried to see if it's because of the loss of expression, there was no current, but even compensating for the expression, there was no currents. So it's a loss of function mutant. Both of them are loss of function mutants. We also looked at the expression of the channel in the membrane, and there was no change in the expression of channel in the membrane. We also performed single channel experiments, and we saw there was a decrease in the P open with both mutants. So it's because of the loss of expression, these mutants are loss of function mutants for GERK channels. And we used GERK openers specific to GERK channels, like VU, it's specific to the GERK channels without GERK1 subunit in them. And ML is a GERK opener, which is specific to the GERK combination where there is a GERK1, like GERK13 or 12. here any homomer is activated by VU. So we used VU to rescue the activity of GERK2 that was lost because of the presence of these mutants, and we saw it nicely rescued the activity. And even for GERK1/3, the ML rescued the activity. So in summary, for K78R, because of the gain of expression, there's a lot of protein, but at single channel level, there is a loss of function property. But overall, there is a gain of function. And we suggest we should use inhibitors in this case. In the other case, where there are loss of function mutants, we suggest we should use activators. So these differential effects of these mutants suggest that we cannot give a generalized medication to all the patients without knowing the molecular mechanism for each mutation. So it urges further personalized medication. If we also have mouse models recently for these 3 mutations. There's a different mutation. These 2 are being under characterization now. So these mice have a developmental delay, and also they show epileptic EEG. And when these mice were treated with ethosuximide, the epileptic activity got alleviated and it went back to normal. With the we also it's a K78R it's a gain of function for potassium channel. It's a counterintuitive to see their epileptic activity. So we think it was because of the overexpression of this mutant in the inhibitory neurons. That's what we saw with the neurons. There's large GABA-induced potassium currents in inhibitory neurons. And with the IATT mouse, also this is also still under characterization. We saw developmental delay even in this model, and also epileptic EEG. So we are planning to apply activators to see if it alleviates the EEG symptoms, epileptic symptoms. Yeah. So despite similar phenotypes, these mutants show different effects on the effectors, and yeah, as I suggested, we should, without knowing the molecular mechanism of each mutant, we cannot go for a generalized therapeutic approach. Yeah. So I'd like to thank my professor and lab members, our collaborators, and our funding agencies. Thank you.
We are perfectly on time, so we have time for a few questions in this presentation. Here.
Thank you. Lovely data. I'm struck by that K78R mutation. It's obviously a relatively conserved mutation. You know, lysine to arginine are both positively charged amino acids. Fairly similar space filling volumes.
All of them are conserved, actually. All of them are in the region where they're supposed to go and bind to the effector or other things. So all of them are conserved.
I was about to say, so in terms of their structural significance, they must be incredibly important areas, because even these very, very conserved changes or mutations are still having significant effects upon the activity of the GERK channels. Yeah.
But they were expressing nicely, and the functionality is also fine. Even through computational modeling, when we checked if the mutation is causing any misfolding of the protein, it's not happening. Yeah.
Thank you.
Okay.
More questions? Yeah.
I can shout.
No.
So beta gamma subunits have 2 effects on the GERK channel. One is a constitutive basal effect.
Yeah.
And one is the stimulated effect following receptor activation. And they are different parts of the ion channel that's involved. So do your GNB1 mutations affect both modulations, or are they preferentially affecting the basal over the stimulated, or vice versa?
Both of them.
So both of them affect, even though they're different parts of the channel that are, wow, okay.
Yeah.
Good answer.
Thank you. Any more questions? Okay. So thank you.
Thank you.